In August of 2018, researchers from the Max Planck Institute for Evolutionary Anthropology sequenced the genome of a 40,000-year-old bone fragment found in Denisova Cave in Russia. They found the hominid had a Neanderthal mother but a Denisovan father. To understand how the scientists came to this conclusion, we should know a bit more about Neanderthals and Denisovans. Neanderthals and Denisovans are two of our closest extinct evolutionary relatives. Around 300 to 400,000 years ago, a group of Homo heidelbergensis, an ancient human species, moved from Africa to Eurasia. They split, and those that moved west to what is now Western Europe and Asia became Homo neanderthalis, or just Neanderthals. Those that moved east towards Siberia eventually became Homo denisovans, or just Denisovans. The scientists discovered two Denisovan individuals in the Denisova cave that showed evidence of gene flow between Neanderthals and Denisovans. These individuals were named Denisova 3 and Denisova 11. Denisova 3 was the first Denisovan individual to be identified, and small amounts of Neanderthal DNA were present in Denisova 3's genome. A bone fragment was found from Denisova 11. The genome of Denisova 11 showed that the individual was about 13 at the time of death and had a Denisovan father and a Neanderthal mother. The father's genome revealed that he came from a population of Denisovans that were discovered later in Denisova Cave, but it also bore traces of Neanderthal ancestry. The mother's genome revealed that she came from a population of Neanderthals that were closely related to a population discovered later in Europe. The data found from the bone fragment revealed that there were at least two interactions between Neanderthals and Denisovans, the first being between the Neanderthal mother and Denisovan father, the second being an interaction in the ancestry of the father.
Here, in this figure, we can see the geographical locations of various hominid individuals located across Eurasia. They are color-coded by species, with blue representing Neanderthals, red for Denisovans, and yellow being ancient modern humans. Part A of this figure shows the percentage of matching Denisova 11 alleles, with Neanderthal at 38.6%, Denisovan at 42.3%, and modern human at 1.2%. Part B is a comparison of the heterozygosity of each species by measure of transversions, which is a point mutation in DNA where a single purine is changed for pyrimidine. Denisova 11 was found to have 3.7 transversions per 10,000 autosomal base pairs. This number matches the prediction of an individual carrying one set of Neanderthal chromosomes and one set of Denisovan chromosomes. Part C shows the percentage of sites at which two DNA fragments carry two Neanderthal alleles, NN in blue, two Denisovan alleles, DD in red, or one allele from each type, ND in purple. On the x-axis, we see the expectations for an offspring of Neanderthal and Denisovan, F1, two F1 parents, F2, and F1 and Denisovan, F1 by D. The plotted results show an almost exact match between the Denisovan 11 column and the F1 column, suggesting that Denisova 11 was the product of two distinct species and not a population with a mixed gene pool.
The interaction between the Neanderthal mother and Denisovan father that resulted in an offspring is a direct correlation to the biological species concept and gene flow. Gene flow is defined as when one distinct or different population moves to another population and its gene frequencies are intermingled with the population that was already present. The two populations then undergo changes in their genetic diversity. To put it simply, gene flow is when migration and mating occurs that results in two populations becoming more similar to each other. It is evident from the bone fragments in the genome of Denisova 11 that the two populations did experience mixing when Denisovans lived in eastern parts of Eurasia and Neanderthals resided in the western parts of Eurasia. The biological species concept is defined as the ability of two individuals to successfully produce a viable offspring. Even though the Neanderthal mother and Denisovan father were from two different populations, they were in fact the same species. We know this because they were able to produce a child that was fertile. Because of the known populations of mixed Neanderthals and Denisovans, the child was in fact viable.